And welcome, Yusuke. Those of you who've been around for a while will know Yusuke as a former CDU undergrad and honours student. That's right, Keith, isn't it?
Yeah, 20 years ago.
20 years ago?
Yeah.
So you're welcome back. And I've been working with Yusuke for the last 7 or 8 years as one of his PhD supervisors, which started before I came to CDU down in Canberra at ANU, where he was interested in doing a PhD and working out how we could use genetic data to track crocodile movement around the Northern Territory. It's been a really interesting collaboration. He was doing that PhD sort of in conjunction with his work as a croc scientist in charge of crocodile monitoring and research with the Northern Territory Government Flora and Fauna Division. And so I guess thanks to them for supporting you to do the PhD through your work as well. So
that's really very lucky.
Yeah. So it's been a really good collaboration. From my perspective, it's put me in the good books every time the university has anything happening to do with crocodiles. The Vice Chancellor says, oh, we've got Professor Sam Banks, one of the world's experts on crocodiles, which is completely and utterly incorrect. Even Keith's smoking here, because I literally know nothing about them. I just sort of absorbed a bit of knowledge from working with Yusuke along the way, but it gives me some credibility, so there you go. Anyway, so today is going to be talking about movement and dispersal of saltwater crocodiles. And like I said, it's a project that he's done for his PhD, which he recently submitted, congratulations. And it started with the idea of looking at using DNA to track movement of crocodiles around Darwin Harbour and sort of managing the source of problem crocodiles there. But he's really sort of taken a huge amount of leadership of the project, and it's expanded to an international collaboration from Australia right across Southeast Asia, up to places like the Philippines and Cambodia to look at movement of crocodiles on a large scale. And it's interesting project to be involved with, and I'll leave it to you to take the way.
Thank you. Okay, so my project is all about the movement of saltwater crocs in Australia and other countries around Australia. As Sam said, this was also my PhD project, and Craig and Sam were my primary supervisors, as well as a few more, like Keith, Hamish here, and Graham Webb. So, and also so many people have been involved. So we just go through. Okay. So study species, everyone knows that crocs, crocodiles porous salties. They are the largest crocs, extant crocs, so living crocs. Some males can grow over 6 meters, weighing more than a ton. And they are found in both freshwater, brackish water, saltwater, any water. So they are widely distributed in the Pacific, Indo-Pacific region, as we'll see soon on the map. They can be quite territorial or defensive or curious, so sometimes, often, they can be dangerous to other animals, including people and stock and pets. And their skins are commercially valuable. For that, they were in the past over-hunted. And in 1990, they got protected in 1971. And since the protection, they've been recovering quite well here. And in other places as well around the same time. Then this is the distribution. The species range quite large over many countries. The first question for this project was their movement, like at large scale, whether they move between countries or islands and things like that. Crocs are often seen in the blue ocean or beach, so they move around a lot, but we don't know their extent. Now, this is one example, Malaysia and Singapore. And between the countries, crocs almost daily move between these countries, but distance is in this case very short. And this is just an ASEAN tip of the Malay Peninsula. And another example, but not known. This was one of our questions for the project. Between East Timor, Timor-Leste, and Australia, people living in Timor strongly believe that crocs attacking them over there are coming from here. So big crocs, dangerous crocs, swim across the sea and reach Timor-Leste and attack people over there. But it's never been shown, so we wanted to find this out. And I'll show you later on. And also at the smaller scale, in Darwin Harbour, crocs use some particular type of vegetation for making their nests, which is tall freshwater floodplain grasses and sedges. So if you map out the vegetation, you can quite easily predict their nesting habitat. If you do that in the Darwin Harbour, you can see there's not much good nesting habitat in there. But our rangers, croc rangers catch about 250 crocs they remove out of the harbour every year. But we didn't know where they come from, whether they come from everywhere or there's some particular source. So this was another research question for us. So the big aim for this project was to understand crocodile movement patterns around Australia, especially here in the NT and other countries. In particular, in terms of environmental influences on their movement, anything affecting their movement patterns, and also the sources and sinks of migration. So where they come from and settle in. Also connectivity between populations. So to answer these questions, we took a genetic approach with the salmon crab. We used this particular method, that SNPs, using commercial laboratory called DART located in Canberra. So we extensively used their method for genotyping our DNA samples. So we started from collecting the DNA samples from anywhere, everywhere, anywhere possible. So these photos are Darwin Harbour with the rangers catching crocs from the trap. And also croc eggs collected by croc farmers. They were another good source of DNA samples. And so croc babies hatched from the eggs. And this is just a quick video of a biopsy. So we used the biopsy as well to collect DNA samples. This is in Kakadu, and with a biopsy pole, aluminium pole with a little needle attached to the end of the pole. You just poke into their tail, like mosquito. Thanks. And when you poke in their tail, they just jump up and go away, swim away, even these big ones. And we keep skin tissue in ethanol in a little vial, plastic tubes, and we send them to the lab. So for Australia, we analyzed just over 1,000 DNA samples, mostly from across the NT, but we also got some from WA and Queensland. Thanks to their governments there, we had good collaborators, so they let us use their samples as well. So first we analyzed these Australian samples, and this is the first result we found out. So there are some geographic barriers like Quebec Peninsula that can prevent the movement of crocs. So here in these graphs, different colors mean different genetics of crocs, so like populations. So here, yellow bit, they are from animal, and here is Darwin or crocodile. So this side is blue and green, and this is yellow. So you can see quite separate by colors. So this is the genetic. Then also we were fortunate enough to analyze some historical data from satellite tracking that was done back in 2005 by Parks Australia Kakadu people and the Crocodiles Park, so Graham Webb and his people. And they did some interesting experiment. So they caught a few crocs, for example, the top one. They caught one croc in Blythe River in Animaland. Then they transported him and left him out at the Mary Shady Camp. Then tracked where he went. And this is the detail of his tracking. Once he was out, he tried to go everywhere looking for his home, but he didn't quite make it, and he finally settled back into Sampan Creek Shady Camp. So he made it his new home. And another example is here. So again, they caught a crocodile at King Gumadia River. So just on the Animaland side, and they brought him back to Kakadu West Alligator River, let him out in the river mouth. Then again, he tried to go everywhere, but in the direction of his original place. He made it pretty close, but he didn't quite make it. He didn't quite make it because inland, these two rivers are not connected. So at the end, he gave up and just settled in there too. So again, Quebec Peninsula here is being a big barrier. Both of them didn't try to swim around the peninsula to go home. And we had a few more examples just like this, well, the other way around. And none of them made it around the peninsula. So this is a big barrier for crocs in MT, separating them into two big groups. So we published these results in one paper back in 2019. So if you are interested in more details, you can check this out. Then the next finding is, as I showed you just now, crocs, despite previous hunting, our crocs, our populations tend to retain relatively good genetic diversity. So again, different colors, different genetics. So sort of quite good separation. So for example, here's a Davie River in the west, and it's red. And next one is still same color, and that's Finesse and Reynolds River. A bit more yellow as you go to more east. Then this blue is TV Island, so here. So TV Island crocs are very different from other crocs in the rivers. So good separation between the populations, which is quite nice to see. And I borrowed this diagram from the report by the Queensland government here. Our croc population here in the NT, we think there are 100,000 crocs, so big mob here. And then, sorry, I should mention this one as well, sorry, quickly go back. We also found that about 42% of crocs on average in any population here migrates from other rivers, but within 200-kilometer range. So they come into new place, but not from far, within 200. So as I said, Daly River, Reynolds, Finesse, they're pretty close within 200 kilometers. And here as well in Arnhem, a bit. So crocs move between these rivers in each region, but not they jump far over the regions. So again, big population here in the NT, but 42% are migrants from 200-kilometer range. In comparison, in Queensland, their population is much smaller, 30,000, and only 10% of them are migrants from 50 kilometers, so within much shorter distance. So that means our crocs are much more mobile comparing to Queensland. And then next one is, so again, we did more detailed analysis and it supported mainly crocs. So in any one population, half of them are locally born in the river, but another big mob came from within 200-kilometer range. But also quite a few came from really much farther, but it's not many really, but up to 700 kilometers. So this is just trend, it's not really definitive. So, and we also found out there are dispersal patterns, so whether they move short distance or long distance is pretty much influenced by the habitat quality. So what's available where they were born and what's available where they settle in. One thing we found out is populations of rivers with a big breeding area, like Arafura swamp, they pump out lots of new crocs, so young crocs. And then they go everywhere looking for new place, and where they settled in, it's not necessarily always nice area. It's not, but they just take it whatever available. Just like human, we start, making houses in the city. We just go outside. So I think this is part of a process like saturating areas, environment for populations. So we published these results in another paper in 2021. So if you are interested in more details. Then back to Darwin Harbour, where they come from and end up in the harbour being cold. So we did some population assignment. We used a few different methods for that, and they all consistently showed some same results. About half of crocodiles in the harbour came from just east of the harbour. So that's Adelaide River, Erie River, and those rivers in Canada. Half of the crocs came from this area. Now, then 20 to 30% of the crocs in the orange bit in the graphs, they come from the west, so Daly, Finesse, Reynolds. then 10 to 15% from TV Islands. So they were born here and they came into the harbor. And very few from animal around the peninsula, but not many, because this is a big geographic barrier. So that's quite interesting. So then be more details about harbor crocs. So again, mostly they came from within 200-kilometer range. So here, and these crocs included both males and females. Also, you can see a few came from much farther, so like Arnhem, a very few. Then if you look at here, so long distance travelers, again, both included both males and females. And they were between 2 and 3 meter long crocodiles. So they were not huge animals like over 4 meters, and also they were not little ones, so medium size. So again, we published this in another paper just last year, so if you're interested. Then out of Australia, their movement between countries. So thanks to our many collaborators in the other countries, we got a few more samples from other countries. And also the museum, especially Queensland Museum. So this is just preliminary results, but when we map out their genetic distance between populations in different countries, you can see here NT crocs, Western Australia crocs, pretty close, genetically pretty close. And then we have Queensland. Then overlapping with a bit of Queensland, here's New Guinea, south of New Guinea. And this is quite understandable because you have Cape York and Strait Islands, then you have New Guinea. So they move between them, just like using the island as a jumping stones. Then you have New Guinea North, and North is a bit far apart, and that means crocs in the south of New Guinea not necessarily move to North or vice versa. And then all other countries from Asia, they are one group close to each other, but pretty far from Australia or New Guinea. So we can put them into 2 big groups of Oceania or Southeast Asia. So this is the first finding for this international scale. Then Timor-Leste. So over the last few years, we collected quite good number of samples from Timor, 27 or 28, I think, across the country island we tried. Then when we mapped out their genetic distance, then again, here's Timor, Australia, pretty far. So that means probably most likely crocs don't move between here and there. So you think crocs attacking people over there, unfortunately locals, they were born there, not here. And then another analysis, we just did a bit of prediction of migration areas and barriers again. Just like where crocodiles are likely to move or likely to avoid. So brown areas, that's where crocodiles don't use to move. And blue areas is the corridor. This is just prediction based on the genetics. Then again here, NT and Timor, it's big brown, so there's some barrier here. And we have big space here and here. And this is because we simply don't have samples. So in future, hopefully when we get a few more samples here, then we can expand our prediction. But at this stage, what I thought was interesting is these migration areas and barriers roughly match with water depth in the sea. And crocodiles, you can see, tend to move across deep water in the sea. And it makes sort of sense. Water can be really rough in the deep water, so the crocodiles avoid it. They prefer using shallow water because it's safer for them. Again, Timor and Australia, you can see it's pretty shallow, but still they don't move. There could be something else like sea currents or something, but we don't know still. Yeah, this is one thing. Now, we run another program to look at more historical side of their DNA. So ancestral population clustering. So it's just like dividing the DNA samples into ancestral groupings. So it's like genetic populations again. And the top one, again, suggested strongly and crocs can be divided into two big groups, which is Australia, New Guinea, and Southeast Asia. So this follows the biogeographic lines, like walrus lines, river lines, or whatever, but with the different names, it's really the same thing. And you can go further, dividing them into smaller groups, like 5 groups, 10 groups. If you divide them to 10 groups, you can see the original populations that we collected, so different countries. Again here, Australia, New Guinea, and those Asian countries. And likewise, we just run different model program to make a population history tree, just to see how crocs got separated into different countries or populations over time, over long time. Then again, Australia, NTWA, Queensland, and we have New Guinea South, which is closely related to some of the Queensland groups. Here to North, New Guinea. And Timor is somewhere between Australia, New Guinea, and the Asian countries. But again, other analysis showed that Timor is more related to the Asian countries rather than Australia. Interestingly, this analysis picked up one recent migration event, which is this red line from New Guinea north to the Philippines. And then one of our collaborators in the Philippines told us that when they started croc farming in the country back in the 80s, they collected stock from other places, including New Guinea. So that's quite interesting, and it's got picked up by this program. So we hope that we hope to expand our analysis. So we need more samples from more places, including Australia and other countries. With more samples, we can do more things like more accurate population assignment. At the moment in the NT, we can, if you give me one crocodile with unknown source where it's come from, we don't know. Then we can tell roughly where he came from, where he was born at a regional basis. So somewhere Adelaide River, Mary River, or Daly River, or Animal, but not river by river. And with many more samples, we might be able to, hopefully, or maybe not, but that can be useful for like forensics, for example, and other things as well. And also we can build more robust population models, like we constantly get asked how many crocs are there in this river or in the territory and stuff. And we can make the model more spatially explicit, then we can be more confident about our estimate about on crocs. So, and also, as I just showed, this will be pretty interesting for taxonomy as well. This is just one single species, crocodiles porosus, but within that there are a few different groups. In old days, you could call them subspecies maybe, but nowadays people call it more like evolutionarily significant units because they've been unique in their history. They're still same species, but quite unique in their genetics. And we haven't covered the entire range at all yet. So the next 5, 10, maybe 20 years, we will have more answers. So yeah, and at the end, I had lots of collaborators and supervisors, so particularly these people and these groups helped me a lot. So thank you very much to them. Yeah, that's it. Thank you.
Yeah.
We got for questions.
Well, you're right on time. We've got another 10 or 15 minutes for questions.
Good.
I was just wondering if you had any thoughts on the differences in the mobility between the Queensland crocs and the Northern Territory crocs. Is that population density related?
Good point. Actually, we did our analysis, NT analysis first, then they did their own, and only recently they published their results. So maybe next step or at some stage, we can address that question. What makes the difference? Yeah.
But the discussion with them though is that it was far more discrete patches of crop habitat quality with sort of fairly large areas in between of not totally unsuitable habitat, not great habitat, except for sort of up around Western Cape York, as you'd sort of expect, I think, but particularly that eastern seaboardy kind of area of Queensland just have those, a few big rivers with good habitat sort of around the coast there.
But also if the population is 3 times bigger in the NT, there might be population pressure.
Yeah.
No one's to disperse.
Yeah, because it's so patchy, it might be more riskier, I don't know, for crops to move between places in Queensland comparing here. Sorry.
The term problem crocodile, is that just any crocodile that's happened to be in the harbour?
Or anywhere that are considered posing any risk to people and the stock.
So it's just they're there. They're not a problem. Their problem is that they are there. Is that right?
Yeah, I just followed the definition given in the management program, and any crocs that are considered risk to the public. So in the Darwin Harbour, because there are so many people, just any crocs being there is considered a risk, I think, even little ones.
The 250 crocodiles were removed from the Darwin Harbour just last year, a year.
Yeah, every year, every single year.
So you can understand why Timor-Leste then says that the crocodiles are coming from Australia and causing a problem, because we're using the term problem crocodiles. And we've got the money to move them, but Timor-Leste is not going to have the money to move them. So I can understand why they have this myth that they would come from here.
Yeah,
Because we're almost calling them problem crocodiles, and we're not knocking them off. We're letting them go off to Timor-Leste in a way. I know we're not, but that almost looks like as though.
yeah. Some
Why
people,
don't we live with them here? Why is there an attitude that they all need to be removed?
I don't know. You need to talk to the government.
It's sort of a question, actually, isn't it?
And I think that, in US, Brown would know, but they call them nuisance alligators rather than problem. Yeah, different terms. And also one thing to add. In Timor, things are more complicated, so they have cultural attitudes towards crocodiles as being their ancestors and stuff. So it's really difficult, very different from here.
Of course, we have people here who consider the same.
Yes, that's right. So sorry, which was first?
The Sahu Sunda divide is a well-known biogeographic divide, so that's not surprising.
Yeah.
Have you thought about doing some phylogenetic analysis? That would be very easy to prove in terms of the Indonesian proof line going through the Timor Trough there. You'll get a divergence around 18,000 years.
Yeah, I haven't, but I'd like to do it. Yeah, that's good point. So,
I found the 42% wanderers, wanderers fascinating.
Yeah.
In salmon, it's about 15%.
mm.
But I was wondering, is anything known about any phenotypic differences, behavioral or morphological or whatever, between the explorers and the homebodies?
Yeah, that's another good point. I haven't, but I'd like to do. Because there might be some difference in their behaviors. Yeah, like you said.
And because that could potentially point to the drivers to, because I mean that split behavior exists from Drosophila upwards, right?
Yeah.
So it's there in all of them, and you can often find that.
And some old men say crocs from this river are more aggressive, blah, 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 things. So that could be true.
Yeah.
And this 42% of migrants is just on average approximately. So in one population, it could be 50-60%, and another one 20-30%. But when you average it, it's 40.
I was going to ask about the post-occipital scutes. Have you noticed that in certain of the areas with different genetics, that has been different at all? Because I know that's one of the most important physical differences.
And in my observation, it's like Australia and Papua New Guinea and Timor crocs didn't have any extra row of osteoderms. Unlike those like in India and Sri Lanka, they had another line clearly. But someone showed me a photo from Crocosaurus Cove, and one of those small ones, they had an extra line. Right?
Interesting.
Yeah, but that's one particular morphological difference between the regions for sure. It's not 100% all the time, I think. Yeah.
Alton, if you have a question.
Keith got it.
Yeah.
You can send me email anytime. So.
I was curious though, for your future separation of trying to identify what rivers they were in. Have you thought about combining isotopic? I mean, I know it'd be really hard to separate like the alligators from east-west or south and east, but you could maybe use isotopic signatures sort of thing.
Originally, we were thinking about combining DNA and isotopes when we started this 10 years ago. But then we decided that this is too hard, let's just stick to DNA. But in future, isotope stuffs like Hamish groups to do. It's quite interesting and can be useful together. So
The crocodiles that would have to be transferred from somewhere else, that seem to try to want to go home.
yeah.
Then we're sort of talking all the time about crocodiles wanting to move to new locations. It seems to be as though there's sort of a strange sort of difference between wanting to go back to their territory. Is this because they're a certain age and therefore then they, you know, I mean, is there an age, all those ones being transferred, are they a certain length or a certain?
Yeah, that's a good question, and it's not really clearly known. But our observations in the field as well as in captivity suggest that even hatchlings, they like to going back to their place, stick to one place. They establish their territory, even babies. So it can be strong as they grow, but it's already there.
And was that true with males or females, you know, that wanted to go sort of return to their area?
Both have them.
Both,
Yeah, male might be stronger, I don't know, I can't say that, but at least both of them have it. Like even females, they come back to the same site for nesting over many years. So, and they compete against each other for good nesting sites. So, yeah.